It collapses, all the beta cells die, and the mice turn hyperglycemic. And this happens 3 to 4 weeks after transplant. So the question we asked is, if we put human islets into these hyperglycemic mice, will we change the ability of these human beta cells to proliferate and regenerate? And the answer was we did. So basically we put human islet grafts into these animals at a level that wouldn't correct hyperglycemic state, either into a normal glycemic mouse or hyperglycemic mouse. We then let the mice go 4 weeks, also adding a compound called BRDU, which will be taken up by cells undergoing division or DNA synthesis, which we can then identify by staining. We can then visualize the insulin-positive beta cells in red and look for dividing cells in green. Basically, under normal glycemic conditions, you find no beta cells which are dividing, but under hyperglycemic conditions, we actually find a 10-fold increase in the number of human beta cells which are dividing in this in vivo test tube. So now, as you can envision, this same model can be used to test a number of therapeutics or drug interactions to see if we can enhance human beta cell regeneration and proliferation in this mouse model. So in the last few slides, I'm just going to describe how we're trying to recreate the human disease process fully in these animal models. So you need to think about whether the key populations in these mice are in humans and mice for the development of diabetes. And there's really 3 key components: the beta cells, the immune system that comes from the bone marrow, and also the thymus, which is important for T-cell education. So some of the first model I'm going to talk about is ways we are now engrafting human immune systems into these mice and getting them to attack a humanized, genetically, mouse beta cell. So for this, we're using our NSG mice, but these mice now express new human transgenes. In these cases, MHC, which present antigens to T cells. We then take these animals and we match up them genetically with human donors. And the idea being, we want to get human T cells to attack the genetically humanized mouse beta cell. And again, we're working with the same kind of strategies where we're taking T cell blood or spleen from TBD donors and putting those cells into the mice with these genetically humanized mouse beta cells. We have a number of these animals available to us, expressing a number of human transgenes. Again, these were all created recently with funding from JDRF. So the basic experiment, I'm going to show you one of these, is we take T cells from a diabetic donor, genetically matched with our mouse recipient for this A2 MAC allele. We inject them into the pancreas, we wait over time, and then we monitor blood glucose. We look at how the human cells do in these animals with the engrafted at very high levels. We look at pancreas histology and look for autoantibodies. I can admit at this point, these models have not progressed to the state where we can get the killing of the complete mouse pancreas. We see no hyperglycemic states in these animals. What we do see resembles a periencephalitis at this point. So here we're looking at the mouse pancreas, so it's been genetically humanized. In green is mouse insulin, and in red is human CD45, which is a marker of human immune cells. So you can see these human immune cells are now surrounding, these T cells basically, are surrounding the mouse islets. And now we're currently initiating studies to try to figure out ways in which we can get these human immune cells to now attack fully that genetically humanized mouse beta cell. So that's the next step in this model. What this really does, though, by putting in PBMCs or spleens from diabetic donors, we're only looking at the effector phase of the disease. The diabetic state has already been established. And what we really want to do is create a unique model where we can study the full disease process from initiation to disease onset. And for this, we're working with the Harvard Stem Cell Institute, specifically Doug Melton, who's an expert in creating induced pluripotent stem cells. So here's the idea, where we take skin samples from a diabetic or a non-diabetic individual. We would create induced pluripotent stem cells, again, with the capacity to form any kind of cell type, as long as we know how to do it. We would then generate pancreatic beta cells, hematopoietic stem cells to recreate the human immune system, and thymic epithelium, which is critical for the education of T cells. These will then all be engrafted into our mouse models. So now we've created everything we need to recreate the diabetic syndrome: beta cells, immune system, and thymus. This process, this mouse, is still under development, and our colleagues at Harvard are in the process of creating all these 3 cell types. But we have all the unique mouse models now ready to go, where we can graft these in, test functionality, and monitor them for disease progress. So we can study the functionality of these cell populations, disease progress, and ultimately test treatments and cures, and even initiating factors in these animal models. So just to summarize, I've shown you we've generated humanized mouse models to study human beta cell function and homeostasis. We can study these under a number of conditions: euglycemia, hyperglycemia, insulin resistance, and we can study iPS cells and ES cell-derived beta cell-like things in these animals. We're trying to recreate type 1 diabetes in the mice. where we can study the effector phase, really with T cells, and create the entire initiation through effector phase with our IPS approach. And finally, just leave with this last thought. These models are not perfect yet, so we're still trying to develop them and push them further. So we're continuously improving these recipient mice to optimize human cell engraftment and function. And we're doing this by genetically introducing new human growth factors and cytokines to improve beta cell engraftment, survival, human immune system development, as well as further impairment of the mouse immune system. It's not completely gone, so we think by further suppressing that, we'll make this model even better. So it just shows everyone that contributes to this work. We have a big lab at UMass, everyone dedicated to creating these diabetic mouse models to study diabetes. A number of other collaborators at UMass, our great collaborator at Jackson Labs, Linda Schultz, and the Harvard STEM Institute, who we're working closely with for that IPS model. Thank you.